Good morning, everyone.
Good morning.
Good morning.
Good morning. Happy
Good
Monday
morning.
to you. Okay. Well, it is 8 a.m., so I'm going to get started, folks. Welcome back. Hope you had a nice weekend. Hope you had an enjoyable time, family and friends. We're going to start chapter 3 today, okay? So we're moving right along. Just taking a quick look at the course syllabus. You are about 1 week away from your very first lab exam of the course, and you are about 3 weeks away from your first lecture exam for the course, right? So very quickly, we're going to get into the thick of things, so to speak, and you'll be taking some very important exams for yourself. So hang in there. Let me know how I can help you along the way. Do you have any questions at this time before we get started? Okay, not hearing anything, then we're going to begin our conversation with chapter 3. The title of chapter 3 is the cellular level of organization. So chapter 3 is all about cell anatomy and some of the major functions and processes that cells carry out during everyday living activities, okay? So today I'm going to start by showing you a video on cell anatomy. I think the video I plan to show you is this one, Biology Cell Structure by a company called Nucleus Medical Media. And this is a very nice video that talks about prokaryotic and eukaryotic cells. Prokaryotic cells would be things like bacteria. Eukaryotic cells would be things like plant cells and animal cells. So it's a really good way to introduce some of the major talking points of chapter 3 in your textbook. Okay, I have already placed the video link for the video into your class notes. So if you miss any part of it here this morning, you'll be able to review it during your own private study time. Okay, we're going to get started here. So just enjoy this as a way to introduce the topic to you. And here we go.
Cells are the smallest living units of an organism. All cells have 3 things in common, no matter what type of cell they are. All cells have a cell membrane, which separates the inside of the cell from its environment, cytoplasm, which is a jelly-like fluid, and DNA, which is the cell's genetic material. There are 2 broad categories of cells. The first category is eukaryotic cells. They have organelles, which include the nucleus and other special parts. Eukaryotic cells are more advanced, complex cells, such as those found in plants and animals. The second category is prokaryotic cells. They don't have a nucleus or membrane-enclosed organelles. They do have genetic material, but it's not contained within a nucleus. Prokaryotic cells are always one-celled or unicellular organisms, such as bacteria. So, what are organelles? Organelle means little organ. Organelles are the specialized parts of a cell that have unique jobs to perform. Let's start with the nucleus, the control center of the cell. The nucleus contains DNA, or genetic material. DNA dictates what the cell is going to do and how it's going to do it. Chromatin is the tangled, spread-out form of DNA found inside the nuclear membrane. When a cell is ready to divide, DNA condenses into structures known as chromosomes. The nucleus also contains a nucleolus, which is a structure where ribosomes are made. After ribosomes leave the nucleus, they will have the important job of synthesizing or making proteins. Outside the nucleus, the ribosomes and the rest of the organelles float around in cytoplasm, which is the jelly-like substance. Ribosomes may wander freely within the cytoplasm or attach to the endoplasmic reticulum, sometimes abbreviated as ER. There are two types of ER. Rough ER has ribosomes attached to it, and smooth ER doesn't have ribosomes attached to it. The endoplasmic reticulum is a membrane-enclosed passageway for transporting materials, such as the proteins synthesized by ribosomes. Proteins and other materials emerge from the endoplasmic reticulum in small vesicles, where the Golgi apparatus, sometimes called the Golgi body, receives them. As proteins move through the Golgi body, they're customized into forms that the cell can use. The Golgi body does this by folding the proteins into usable shapes, or adding other materials onto them, such as lipids or carbohydrates. Vacuoles are sac-like structures that store different materials. Here, in this plant cell, the central vacuole stores water. Going back to the animal cell, you will see an organelle called a lysosome. Lysosomes are the garbage collectors that take in damaged or worn-out cell parts. They are filled with enzymes that break down this cellular debris. The mitochondrion is an organelle that is the powerhouse for both animal and plant cells. During a process called cellular respiration, the mitochondria make ATP molecules that provide the energy for all of the cell's activities. Cells that need more energy have more mitochondria. Meanwhile, the cell maintains its shape through a cytoskeleton. The cytoskeleton includes the thread-like microfilaments, which are made of protein, and microtubules, which are thin, hollow tubes. Some organisms, such as plants that are photoautotrophic, meaning they capture sunlight for energy, have cells with an organelle called a chloroplast. The chloroplast is where photosynthesis happens. It's green because it has a green pigment called chlorophyll. Plant cells also have a cell wall outside of their cell membranes that shape, support, and protect the plant cell. Animal cells never have a cell wall. There are many other unique structures that only some cells have. Here are just a few. In humans, for example, the respiratory tract is lined with cells that have cilia. These are microscopic hair-like projections that can move in waves. This feature helps trap inhaled particles in the air and expels them when you cough. Another unique feature in some cells is flagella. Some bacteria have flagella. A flagellum is like a little tail that can help a cell move or propel itself. The only human cell that has a flagellum is a sperm cell. In summary, remember, eukaryotic cells are plant and animal cells with a nucleus and membrane-enclosed organelles, while prokaryotic cells are unicellular organisms without these things. All cells have a cell membrane, cytoplasm, and genetic material. And even though only plant cells have chloroplasts, both plant and animal cells have mitochondria.
Okay, folks, so there is a quick introduction to the major talking points of today's class with you. Okay, and so let me bring up your images again. Okay, did you have any questions from watching the video one time? You know, when you look at some of the interior of a cell, it's kind of like you were transported to a foreign planet with all these kinds of exotic things in that we've never seen before. Okay, it's something that we don't typically spend our lives thinking about unless you're a biology teacher. Okay, so any thoughts or questions before we continue? Okay, all right, well, let me know. All right, we're going to start introducing you to the major anatomy of a typical human cell, which is basically what is covered in chapter 3 of your book. If you want visualization right now, you could be looking at figure 3.1. Figure 3.1 on page 64. It gives you the typical structures found in body cells. Let me start by emphasizing once again that there are two major types of cells on the planet, two major types of cells that we've discovered on planet Earth. And they are prokaryotic cells. Okay, examples, prokaryotic cells would include bacteria. It would also include some other things that I'm not going to tell you about right now, and be happy to tell you about someday if you take a microbiology course. But bacteria would be what you are probably used to thinking about. You've heard of bacteria. You know that sometimes you take an antibiotic to treat a bacterial infection. So bacteria are prokaryotic cells. And when we say that it's prokaryotic, we mean that it does not have a nucleus. There's no nucleus present. Okay, so an example of a bacterium would be the bacterium Escherichia coli, or E. coli for short, which you know is commonly found in the large intestine of your body, right? It is a natural inhabitant of our bodies. Okay, yes, Brittany, I'm on figure 3.1, which in my textbook, I'm using a printed textbook, so my page numbers might be a little bit off from your digital version of the textbook. My page number is 64, but it's figure 3.1. Okay. All right, so E. coli is an example of a type of bacterial cell. Bacteria are prokaryotic cells. They do not have a nucleus. And you say, well, that's not important for me in anatomy and physiology, but it really is important, because think about how often our health is changed, altered, made better, made worse by the presence of microorganisms that are in our bodies. Okay, the other major type of cell is eukaryotic cell. These do have a nucleus. I'll just say it this way: nucleus present. This would include plant and animal cells. Okay, obviously the example that we're thinking about are cells of the human body. Okay, and really that's where we're going to make the emphasis of our conversation today. Okay, so as I go through my list of anatomical structures, you can continue to look in figure 3.1, page 64, or you can also highlight the structures with different pictures, and I'll try to reference them with figure numbers during our conversation. Okay, so we're going to do a cell anatomy tour of a human cell. Okay. The very first thing is to talk about a structure called the plasma membrane. Plasma membrane is shown to you in a magnified, detailed view on figure 3.2. Okay, and so what I'll just do is highlight some of the anatomy of the cell, maybe give you one or two talking points about it. All we're doing is introducing these things together at this time. We could have a semester-long seminar or entire course in the plasma membrane. We could have a semester-long course in any part of the cell, because it's that important. There's so much to know about all of these things. Okay, thank you for the comment, Brittany. Okay, so the plasma membrane is really the border of the cell. It is this physical structure that defines the inside of the cell versus the outside of the cell. Okay. This is the physical border of the cell, which defines the inside and outside of the cell. Okay. It's kind of like, if you think about your house, it's kind of like your exterior wall, right? The exterior wall of your house is the border that defines what is going to be considered inside your house versus outside your house. That is the plasma membrane. Okay, the plasma membrane is made of the following. Predominantly, it is made of phospholipids. If you look at figure 3.2 on the bottom left corner of the image, you see phospholipids. And notice that there are two layers of phospholipids, which is why it is described as a lipid bilayer or a phospholipid bilayer. They're actually two layers of lipids that are oriented with respect to each other, tail to tail. Tail to tail when we think about phospholipid structure, which you would be studying from chapter 2. So it's made of a phospholipid bilayer, and it's made of additional molecules that are actually interacting with the phospholipid bilayer in some way. As you look across the image in figure 3.2, do you not in fact see these purple structures, which are different kinds of proteins, right? So there's other molecules sticking through and hanging off the inside and outside of the phospholipid bilayer. Okay, some of these molecules will include proteins. Okay, and I won't go into details just yet, but on figure 3.2, do you notice the integral proteins, also known the peripheral proteins that hang on the outside and inside of the bilayer. So there's many proteins sticking through the phospholipids. There's also what we call as glycoproteins, right? The animation I showed you a protein with a carbohydrate attached to it. That's called a glycoprotein. There are also glycolipids, okay, lipid molecules that are attached to carbohydrates. Those are also part of the phospholipid bilayer. I'll mention one more thing, that if you look very carefully on figure 3.2, bottom left corner, it's a tiny little thing, but it's highlighted there, and it's called cholesterol. Cholesterol is a molecule that is a structural component of the plasma membrane of all of our cells. Okay, so the plasma membrane is actually a fairly complicated structure. Its main component is the phospholipid bilayer, but there are a variety of other molecules sticking in it and through it that enable the cell to interact with molecules on the inside surface of the cell as well as on the outside surface of the cell. Okay, so one final comment here is that the plasma membrane, if I had to tell you what its purpose is, well, its purpose is to act as the structural border of the cell, but there's a secondary purpose here that's intimately related, and that is the plasma membrane's purpose is to regulate what kind of material is able to enter the cell and what kind of material is able to exit the cell. The plasma membrane controls that. So it regulates the entry and exit of cargo from the cell. You think about, typically we're going to eat several meals a day. The nutrients from the food that we've eaten, at least some of those nutrients have to get into our cells so that our cells have their own food to live, right? So that you got to get across the plasma membrane for that to happen. Plasma membrane is going to regulate and coordinate what material is able to successfully enter the cell. Okay, do you have any questions right now? Okay, so we're just going to start rolling here. The next thing to mention is the cytoplasm. Okay, it's not terribly exciting to talk about the cytoplasm. Talking about the cytoplasm inside the cell is kind of like talking about the air in the room you're at, right? Think about it. If you were to reach up your hand and point to any random location of air in the room, you would be pointing to air. It's just everywhere. The air is everywhere. Well, the cytoplasm is everywhere. Okay, the cytoplasm is, in an analogy sense, equivalent to the air in a room. It is the air of the room, but it's not made of air. It's actually made of water and a variety of solutes, which changes the physical consistency of the cytoplasm from a liquid to a jelly. So the consistency of cytoplasm is kind of more similar to the grape jelly you might have in your kitchen refrigerator upstairs than it is water. So cytoplasm is basically all the inside material of the cell that is made of water and dissolved solutes. Okay, it has the consistency of jelly, or you might say the consistency of honey, or the consistency of molasses. Okay. All right. So it's basically the water inside the swimming pool. That's what I'm saying. The cytoplasm is the water inside the swimming pool, except that the water is more like a jelly, not just liquidy water. Okay. Let's continue. The cytoskeleton. Okay. The cytoskeleton is next. The cytoskeleton is something that is highlighted in your book. And, I'm going to be moving around here to show you some different pictures. But let me see if I can get what I need. Okay, yes. Figure 3.15. Figure 3.15, page 80, is your cytoskeleton. Okay. The cytoskeleton is a network of tubes where all the tubes are made from different kinds of proteins. Okay. And so I don't care too much if you know what these different proteins are right now, but I'll point them out to you. In figure 315, there are, three major types of tubes. They are microfilaments, intermediate filaments, and microtubules. I'm not going to go into detail about what the composition of these tubes are at this time, but I'm going to tell you a little bit more about the broad purpose of the cytoskeleton. Okay, so these are the three major types of tubes that make up the cytoskeleton. Yes, they are made of different kinds of proteins. The purpose of the cytoskeleton is twofold. Okay, the cytoskeleton does two major things for us. Number one, it helps provide structural integrity to the cell. It's kind of like the 2x4s in the walls of your house. They provide structural integrity, right? They make sure that the strength of the walls is able to handle the weight you've placed on them, so you can put furniture on your floors and things don't collapse. So cytoskeleton provides structural integrity to the cell. And thirdly, this might seem strange to you, but the cytoskeleton has a network of tubes which span the entire internal dimension of the cell, from one internal edge of the plasma membrane to the other internal edge, right? Like if between my hands I had a ball, and that ball was representative of a cell. if I had a tube that ran between the top of the ball and the bottom of the ball, the whole length, that could be a microfilament. It could be a microtubule. It could be an intermediate filament. Okay, so the cytoskeleton is a network of tubes, and that those tubes not only provide structural strength to the cell, they also act as a road network for machinery inside the cell to walk along the tube like you're walking along the road, moving cargo from point A to point B inside the cell. Okay, so once again, we come to the realization that the interior of the cell kind of looks like an exotic foreign land or a foreign planet that we might be transported to. There's lots of odd things in there that we're not used to thinking about that we need you to start thinking about. Okay. Do you have any questions you'd like to ask? Okay, I'm going to move to the topic of ribosomes, page 83, figure 318. Okay, ribosomes. Ribosomes, folks, we may have mentioned this before in our class. Ribosomes are the nanomachines, right? Nanomachines that the cell uses to manufacture protein. Okay, this chapter 3, towards the end of the chapter, actually explains how cells use ribosomes to manufacture protein. We will be talking about it, not today. This is probably for a next lecture, or even the third one beyond that. Okay, but let me ask you a question in the chat window. Here we go. What is the name of the process cells use to make proteins? What's it called? Does anybody know? Do you have any background in this from, from studying perhaps another course at another moment of your life? What is the name of the process cells use to make proteins? What's it called? Okay, thanks to everybody that is contributing. I really appreciate it. Okay. Protein synthesis is certainly a phrase that your textbooks use. So I can understand why you said that. I'm going to be a little bit more precise from a science perspective. The process is known as translation. Translation is the major talking point of figure 3.28 and figure 3.29. Okay, so we'll be talking about that not today, certainly not today, but another time perhaps. Okay, so let's put that down in your notes. The process of protein synthesis is called translation. The process of protein synthesis is called translation. All right. Let me just mention a couple things here. In terms of, where inside the cell are you going to find ribosomes? Well, you're going to find ribosomes floating just randomly in the cytoplasm, and you will also find them attached to a membrane structure, which is next on our list to talk about, called the endoplasmic reticulum. So some ribosomes are found floating freely in the cytoplasm, and others are attached to a membrane called the endoplasmic reticulum. One of the things I'd like to do is give you a little bit of numbers on things to give you a realization of just how many of these things are present. Okay, a typical bacterial cell like E. coli will have around 50 to 60,000 ribosomes in an individual cell. Now, your animal cells are quite a bit larger, so we can expand that to say that typical number of ribosomes in a human cell is going to be on the scale of hundreds of thousands. Hundreds of thousands of ribosomes are present in a typical human cell. It's difficult to give you an exact number because the number of ribosomes is not constant, and the variety and types of human cells that make up your human body are different shapes and sizes with different internal volumes. So you could have, you might have a small cell in your skin that has 100,000 ribosomes. You might have a giant cell like a neuron, which has a long axonal fiber that's 3 feet long. It might have 900,000 ribosomes. It really depends on the kind of cell we're talking about. But the take-home message here is, unlike what you might think from simply looking at a book, you don't have one ribosome in a cell. You have hundreds of thousands of ribosomes in a typical animal cell. And that tells you that obviously for the cell to have so many copies of a ribosome, that the process that ribosomes perform must be very important to the capacity of a cell to actually be alive. And that capacity is the production of protein, translation. Translation is deeply important to allow a cell to live and to stay alive. All right, let's move on to the topic of the endoplasmic reticulum. It's a logical jumping point from our ribosome conversation. If you turn the page in your textbook, figure 3.19. 3.19. Okay. The endoplasmic reticulum is a network of membranes that extend off the membrane of the nucleus. Okay, a network of membranes that extend off the nucleus of a cell. Now, we haven't typed nucleus into your notes yet, but you did hear about it in the video, right? The word nucleus was present. So you can see for yourself what it looks like in figure 3.19. And if you look carefully, you can see that in fact, the membrane does extend off the membrane of the nucleus. Okay, so there are two major types of endoplasmic reticulum. Okay, they are the rough endoplasmic reticulum, also known, as the video said, the endoplasmic reticulum is sometimes called ER, right? Endoplasmic reticulum. The rough endoplasmic reticulum is called rough because it is one of the major locations you're going to find ribosomes attached. So rough ER is called rough because there are attached ribosomes. The second type of ER is the smooth ER. This is called smooth because it does not have any ribosomes. There are no ribosomes attached to the surface of the smooth ER membrane. Okay, and so that, the difference in naming goes back historically to when scientists first started looking at the interior of cells with microscopes, and they saw these things and they said, okay, where we have these membranes, some of them have ribosomes, some of them don't. So one's going to be called rough, the other's going to be called smooth. Okay, the purpose of the endoplasmic reticulum membrane is to provide a great deal of surface area so that You can embed structures, sometimes ribosomes in the rough ER, sometimes just enzymes. You can embed molecules that then can perform meaningful chemistry. Okay, so the purpose of the rough ER is to create a huge internal surface area for the cell to perform meaningful, meaning useful chemistry. So let me give you an example. Okay, obviously the rough ER has ribosomes. You can make proteins there. You already know that. We've said that. There's something else. Okay, in the rough ER, there's also going to be a variety of enzymes that are involved in the production of phospholipids. So the rough ER has enzymes that make phospholipids. Okay, so phospholipids keeps coming up in our conversation, right? We just talked about it as, the phospholipid bilayer of the plasma membrane, and we referenced phospholipids in chapter 2 as one of the major subtypes of lipids. Okay, so phospholipids are made by enzymes that are sticking in the fabric of the rough endoplasmic reticulum membrane. Just to give you a piece of information about the smooth endoplasmic reticulum. The smooth endoplasmic reticulum contains enzymes that are involved in the synthesis of fatty acids and different steroid molecules. Okay. There's one more thing I'll mention to you about the smooth endoplasmic reticulum. The smooth endoplasmic reticulum is one of the things we typically tell students about. If your organs, if your body is exposed to potentially harmful materials that you've ingested, then your body, the cells of your body will try to detoxify, will try to chemically alter those potentially damaging materials so that they don't harm your body. Well, one quick example of that is alcohol. Now, we drink alcohol for a variety of reasons, okay? But the point of it is, from your body's chemistry perspective, alcohol is really not a desirable substance. And so when you drink alcohol, obviously you might start to get a little tipsy, you might get a little inebriated, but you appreciate if you stop drinking alcohol, you do not remain tipsy, right? Over a period of time, you will sober up. Well, how do you sober up? Sobering up means that the alcohol you've ingested will be chemically modified by enzymes present in the smooth endoplasmic reticulum of your body cells, but most notably your liver. Your liver organ has many cells that are capable of detoxifying substances you have ingested, like alcohol. How does it happen? There are enzymes in the smooth endoplasmic reticulum of your liver cells, of the liver organ, that will chemically alter and change the alcohol into something that's not dangerous. So the smooth ER contains enzymes to detoxify potentially harmful substances like alcohol. All right. Okay, gang, we're moving right along here. Do you have any questions right now? Okay, let's keep rolling. All right. All right, so endoplasmic reticulum was letter E. Let's go to letter F. Okay, letter F is the Golgi complex. That would be figure 320, page 85, the Golgi complex. The Golgi complex is also known as the Golgi apparatus. Okay. In the video you watched, the narrator of the video said that the Golgi complex is also sometimes called the Golgi body. That is generally not correct anymore. Okay, so don't use the phrase Golgi body. Golgi body is a virology term used in microbiology. So Golgi complex and Golgi apparatus would be correct terms. Golgi body would be an old deprecated term. Okay, so Golgi complex is what you see there in figure 320. It also is sort of a network of back and forth membranes that form this membrane structure. Okay, and what the heck is it? Well, it is an organelle that, if I were to apply a single analogy to, acts in a similar manner to your post office. This is the post office of the cell that you're looking at on page 85. This is roughly equivalent to our post office. And by that, what I mean is that this organelle is responsible for modifying, sorting, and packaging materials, particularly proteins that are delivered into it. Think about it. If you have a letter that you want to mail, but you're not quite sure of the address or how to write the address, you might go to the post office and they might help you write the address on the letter, on the envelope. You might have a package that you need to send to give somebody a Christmas present. So you take your package into the post office and they tell you, oh, well, you're going to need this kind of packing material, or you should put it in this kind of box, and they'll help you package it up for safe delivery. That's what the Golgi complex does. Okay, so the Golgi complex modifies, sorts, and packages materials, especially proteins, for delivery to other destinations. Okay, you probably don't know this yet, but you will by the time we're done with chapter 3. The typical workflow of manufacturing a protein involves decoding information in the nucleus, which is then used to build some material called RNA, which is then exported for the nucleus and shoved into the rough endoplasmic reticulum, where ribosomes take the RNA and make that RNA then into protein in the process of translation, which we'll go into later. That protein is then typically delivered to the post office, typically to the Golgi complex, where it can be addressed, manipulated, packaged. And the purpose of doing that is so that machinery in the cell knows where the cargo, the protein, is supposed to be delivered, because some proteins should be delivered back into the nucleus. Other proteins should be delivered to the endoplasmic reticulum. Others should be delivered to the plasma membrane on the border of the cell. And even other proteins should be actually transported out of the cell. We should have it leave the cell and float through the bloodstream to another destination. So the Golgi complex kind of addresses the cargo in a way that the machinery of the cell knows where to deliver the cargo. Okay. All right. This is super important, because you can imagine if, think about our own daily lives. If you pay a bill and you write a check out and put it in an envelope, but you don't get the right address on the envelope, so the bill goes, your bill payment goes to the wrong destination, it never gets to the company. Obviously, you're going to be in default of your loan or whatever it is you're paying. That would create a problem, would it not? Same way that if cargo in the cell does not get to its proper destination so it can do a proper job, a proper function of the cell, then typically things start to break down in the cell, and you might actually create disease at the molecular level. So Golgi is super, super important. Okay, thoughts, questions? Okay, we're just getting started here, gang. Let's continue. Next part of the cell I'm going to talk about are the lysosomes of figure 3.22. Figure 3.22 page 87. Okay. Lysosomes are membrane enclosed structures, membrane enclosed vesicles that are really recycling facilities. That's the analogy I like to use. They are the cell's recycling facilities. Think about it this way. Probably once a week, you put out some garbage so that the garbage folks come along and they gather it and take it away from your house. But there's probably also a recycling truck that comes around and gets cardboard, glass, other things that your township or municipality wants to recycle and reuse. Well, lysosomes are the cell's recycling facilities. Okay. And so, what is the purpose of recycling? It's to break something down and reprocess it and reuse it to build something new again, right? I mean, that's what a cell's going to do as well. If you have an old worn out phospholipid or an old worn out carbohydrate or some kind of molecule that's worn out, or even some kind of larger structure like another organelle, like a mitochondrion, which we haven't talked about yet, then the cell can use its lysosomes to recycle that material, break it down into building blocks, break it down into more primitive substances that the cell can then use to build something new again from the material that was broken down. Okay, so lysosomes have an internal environment that is acidic. So there's a lot of acid inside the lysosome. Makes sense that there would be acid because acids are useful in breaking things down, right? Think about it. You have acid in your belly to break down the food you eat. So cells will contain some acid in the lysosomes to break down things that need to be recycled. So lysosomes have an internal environment that is acidic and also has many digestive enzymes that can be used to break down worn out molecules and structures like organelles. Okay, so we can break these things down and we can use the building blocks of those materials to assemble something new. Okay. Now, there's another interesting feature of lysosomes that often goes untalked about, and I'm going to mention it at this time. Okay, and that is defense. Now when you're out walking, maybe you're out walking in your neighborhood, and so maybe you carry a spray can of mace along because maybe there's there's been some burglaries in your neighborhood, or maybe you carry some pepper spray against dogs that are aggressive, something like that. So the point is that we might take action to defend our bodies. Well, doesn't it make sense that cells need to be able to defend themselves against potential harm? We already know, because of the times we're living in with COVID, and COVID is caused by a coronavirus, that viruses enter cells and cause harm. There's all kinds of microorganisms that can enter our cells and cause us harm. So cells need to have some defense capabilities. And so lysosomes can not only be used for recycling, but it can apply the recycling capacity for cellular defense. Okay. Lysosomes can be used for cellular defense. Okay. And, it might make you smile or think that your teacher's odd, but I'm entirely serious when I say to you, maybe if a burglar shows up at your house, to defend yourself against a burglar, you actually push the burglar into your garbage disposal in your sink and grind them up. And that's exactly what the cells try to do with microorganisms that invade the interior of the cell. The cell will try to to move the virus through the lysosome, or in some cases, other things like bacteria, through the lysosome to try to break the virus or bacterium up and destroy it. That's cell defense. You're using your recycling capability to defend against microorganisms that might disrupt the behavior of a cell and make the cell sick. Okay. Do you have any thoughts or questions? Okay, let's continue. Next structure I'm going to highlight for you. And again, you notice I've skipped some things. It's just because I only have so much time. I want to pick and choose what I spend time with. We need to mention the mitochondria, figure 323, page 89. The mitochondria are the organelles of the cell that are the power plants. And by power plants, I use that word very loosely, but what I'm saying is mitochondria are heavily involved in the production of a form of biochemical energy called ATP. These are the power plants of a cell. Okay. Mitochondria are used to make ATP. You know, ATP is biochemical energy. ATP is short for adenosine triphosphate. Okay, and so we're not going to go into the process of how that works now. You actually will go into that process in AMP2 next semester. Okay, it does involve quite a bit of chemistry. So you have that to look forward to in the months ahead. Okay, so mitochondria are used to make ATP, and the ATP is then used by cells to do a variety of different kinds of work, including simply contracting your muscles so you can wave your arms, play baseball, volleyball, dance, walk across the room, that kind of thing, right? ATP is the biochemical currency of the cell. It's kind of like the dollar bill, where we exchange dollar bills to accomplish something in society. If I want a pack of gum, I give a dollar bill, I get a pack of gum in return. If I want to contract a muscle, I give an ATP molecule, it gets broken down, energy is released, I contract the muscle, everybody's happy. Okay, so mitochondria are the power plants of the cell. Much more will be said about them at another time. Again, I want to give you an idea of scale in terms of, well, how many mitochondria do you typically find in a human cell? Again, it varies depending on the kind of cell we're talking about, but the proper scale or magnitude to think about for mitochondria is a typical human cell will have several hundreds to several thousands of mitochondria. Each human cell, depending on the type of cell and its function in the body, will typically have several hundreds to several thousands of mitochondria present. Okay, so a typical human cell will have, 1,000, 2,000, 3,000 mitochondria present, and that same cell will have 100,000, 150,000, 200,000 ribosomes present in the same space, in the same volume of the cell. Okay, so even though cells themselves are very tiny, there's even more tiny material that's present in quite a lot of abundance inside each cell, which allows cells to achieve the work that they have to do. Well, what is the work of a cell? The work of a cell is to live. Even to live requires work, requires expenditure of energy. Okay. I'll mention one more thing here. Mitochondria are intimately involved in a type of cell death process called apoptosis. called apoptosis. Okay. You might appreciate from other biology experiences you've had that, when our bodies were being created, when they were developing, when they were forming inside your mother's womb before you were born, that, for example, you think about your hands, think about your fingers, right? That before you were born, each finger of your hand had some tissue in between the digits, as though your hand looked kind of like a duck's foot, where there was webbing in between the digits of the fingers. Okay. And so obviously, if everything went proper during development, when you were born with individual fingers, where there was no tissue in between the fingers. And you ask yourself, well, how did the body get rid of that tissue? Well, that tissue was programmed to die. It was programmed to die in a process known as apoptosis, which requires the mitochondria to basically stop producing energy to actually trigger a cell death pathway. Okay. All right. Moving on. Next structure we need to talk about is the nucleus. Okay, the nucleus as a topic has text on the bottom of page 89, but then we have pictures, figures, diagrams of the nucleus, figure 3.24, page 90. Okay. The nucleus. Okay, the nucleus is a membrane-bound organelle, which is the primary location of most of your cell's DNA. Okay, a membrane-bound organelle, which is the primary location for most of your cell's DNA. Okay, and these several pages we're on at this time talks about that, tries to give you information about it. Okay, so what can we say here that might be interesting to you? Okay, most human cells of your body are going to have a single nucleus. Okay. Most cells in your body are going to have a single nucleus. Okay, so your skin cells on the surface of your arm, one nucleus. Kidney cells making up your kidneys, one nucleus. Thyroid cells making up the thyroid, one nucleus per cell. But there are some notable exceptions. Okay. Exceptions exist. So, for example, skeletal muscle cells can have multiple nuclei. In fact, they almost always do have multiple nuclei. And we'll talk to you more about why that is when we get there. That's a topic we're going to cover in A&P 1. So most cells in your body have a single nucleus. Skeletal muscle cells are an exception. They have multiple nuclei. Here's another exception. Mature, and I emphasize the word mature. Mature red blood cells have no nucleus. Okay. And this is something that you'll get more in A&P 2, but if you think about it, the purpose of a mature red blood cell is simply to deliver dissolved gases throughout the body, oxygen and carbon dioxide. Mature red blood cells don't actually reproduce, so there's really no reason for them to carry around genetic cargo. Okay. All right. So the nucleus is where most of your cell's DNA is found. The video we watched this morning mentioned a subsection or a subregion, a sub-area inside the nucleus called the nucleolus. Okay, that was figure 3.24. On figure 3.24, you see the nucleolus there. Okay. The nucleolus is a sub-region inside the nucleus involved in the production of ribosomes. Okay, so the nucleolus is a region, it is an area within the volume of the nucleus that's involved in making the building blocks of ribosomes. Okay, most books will tell you, most books will give you the impression that there is one nucleolus in a nucleus. And that is also not really true. You can have multiple nuclei inside a single nucleus. Okay. All right. Okay. All right. Now, I feel like I should talk a little bit, just a little bit about the information on page 91, where it talks about the DNA and the chromosomes. So we'll put that in here with our notes with the nucleus, because that seems to be the most appropriate cellular location to talk about this content. Okay. All right. So most of the DNA inside your cells is found in the nucleus. It's not found as naked DNA, okay? Most of the DNA inside the nucleus is found complex with special proteins called histones. And you can see that on figure 3.25. So the proteins called histones and the DNA form these structures called chromosomes, which you also see in pictures A and B of figure 3.25. Okay, so the DNA inside the nucleus is organized with proteins called histones. To form structures called chromosomes. Okay. All right, I have a question for you in the chat window at this time. How many chromosomes are found inside a typical human cell? Okay. I appreciate everybody talking about this. And if you look carefully in the chat window, you can see there is a disagreement. Is there not? There's a disagreement amongst you. Okay. So we have to be very careful about how we communicate this. What is a typical human cell? A typical human cell is a human cell that's diploid in nature. Diploid means there's two sets of chromosomes, one set that was inherited from your mom and the other set that was inherited from your dad. Okay, so a typical human cell will have 46 total chromosomes that are organized as 23 pairs, 23 groups of two. Again, 23 individual from mom, 23 individual from dad. Okay. A typical human cell will have 46 total chromosomes organized into 23 pairs. Okay. Half from mom and half from dad. Okay, so we really are a hybrid. We are a mosaicism of our genetic material from mom and dad. And there's other things that need to be considered there as well for another time. Okay, so when we talk about this material, you might listen to the evening news, or you might read a magazine in your doctor's office or dentist's office while you're waiting to go see the dentist or whatever. And it might be something where it's talking about science and DNA and stuff. And it's very common to hear the phrase genome, the human genome. Well, exactly what is that? Okay. The human genome refers to the genetic material of a haploid cell. A haploid cell, okay? So a haploid cell is a sperm or unfertilized egg cell, meaning that haploid cell, the sperm or unfertilized egg, is only going to have 23 individual chromosomes, 23 from mom or 23 from dad. The only cells in our body that are haploid are the sex cells, right? The sperm and the egg. Those are the only cells that are haploid. By far, most of our body cells are in fact diploid, which is the 46 here, okay? Most human cells have 23 pairs of chromosomes, which means the cell is diploid. Diploid, again, referring to two sets of chromosomes. Two sets of chromosomes. Okay. All right, so Kendall, that is correct. Kendall has a question in the chat window. So gametes are the only haploid cells. That is correct, Kendall. Gametes are the only haploid cells. Every other cell of your body is typically going to be diploid, barring any other, barring any unusual circumstances that we could talk about at another time, like cancer. Cancer throws the wrench into things. So here's the question that I'll sort of ask it, but then we'll answer it here, okay? Let me ask a question in the chat window. What is the basic building block of DNA called? What is the basic building block of DNA? Now you should be able to answer this because we covered it in chapter 2. What is the basic building block of DNA called? Okay. Thanks, Noah. Thanks, Kelly. Kendall. Okay. Thank you for your participation. I deeply appreciate it. The answer is, in fact, nucleotides. Nucleotides is the basic building block of our DNA. If we think about nucleotides as chemical letters equivalent to the letters of the words in your textbook, the A's, the B's, the C's, A, B, C, D, E, F, G, right? All the letters of the English alphabet. If we think of the nucleotides as chemical letters of DNA, I could ask this question. How many letters are in your textbook? How many letters are printed among all the words of all the sentences, of all the paragraphs, of all the chapters of your entire textbook? How many letters are in your textbook? The answer has got to be a very large number, right? So now I'm going to ask you how many letters, how many nucleotides are found in a diploid typical human cell. Do you have any idea how many letters are in the diploid human cell? How many letters of nucleotides? Okay. So let's put this down as a question here. How many nucleotides of DNA are found in a diploid human cell? Okay, I'm basically asking you how many letters of information is there inside your cells that gives your cells the instructions it needs to carry out life and for you to be the human being that you actually are. Because you appreciate your letters are different from my letters, which are different from your brother and sister's letters, because we're all unique people. So how many letters worth of instructions does it take to make a human being up? Well, Noah, thanks for answering that. No, you are correct that there's 4 nucleotides in DNA, but that's not what you're saying, Noah, is that there's 4 different kinds of nucleotides. What I'm asking is how many total nucleotides are in our human cell instruction manual? The answer is as follows. It's about 6.4 billion nucleotide pairs worth of DNA. Okay, I'm just trying to give you a scale for the magnitude of the quantity of material found inside the nuclei, which is necessary to provide all the instructions to build and maintain the anatomy of your body and all its functions, so that your lungs can inhale and breathe, so that your muscles can contract, so that your brain can think of thought, so that your kidneys can filter your blood to create urine, so that your ovaries can manufacture egg cells, so that your testes can manufacture sperm. You know, whatever happens here, how much instruction is needed? What's the size of the instruction manual? It's 6.4 billion nucleotide pairs of DNA in every one single cell of your body. Okay, so here's a question. How many human cells make up your entire body? Now, there's not going to be one single correct answer because we're all different shapes and sizes. But let's get an idea of the magnitude of scale we're talking about here. Okay, anybody read about this, hear about this from any other moment in life? How many human cells make up your human body? Kendall, thanks for participating. The number is larger than that. Anybody else? Okay, Kelly, you're now starting to get to the correct scale of things, right? The general, yeah, something that needs to be represented with exponents, right? The general scale of things is 50 to 100 trillion. That's the estimate. The average human body is made of 50 to 100 trillion human cells. And you know, that is an estimate because nobody's ever actually counted it, because that would be an insanely tedious job to do that probably could never be accurately done anyway, right? So estimates based on physical properties of the volume and weights of cells is done to estimate the number of human cells that must be making up our human body. Okay. And just for comparison, again, we started today talking about the different kinds of cells on the planet, prokaryotic cells like bacteria. And then we talked eukaryotic cells. We spent all day talking about our human cells. But the reality of it is our health, whether we're healthy, whether we're sick, is heavily influenced by the types of microorganisms, bacteria, fungi, protozoa, viruses that live inside our bodies, and I dare say on the surface of our skin, right? Because the surface of our skin is chock full of microorganisms, whether we like to admit it or not, whether that seems gross or not, the microbes are there and they are contributing positively or negatively to our overall health. So here's the question. How many microorganisms organismal cells are present for each one human cell in the body? How many microorganism cells are present for each one human cell of the body? What do you think? For every one human cell, how many bacteria are present? For every one human cell, how many virus particles are present? For every one human cell, how many fungal cells are present? What is the current scientific estimate on this? Okay, we're just about out of time, so I guess I better give you the answer on this. The answer is 10. 10. Science estimates that for every one human cell in your body, there is at least 10 microorganismal cells in your body. Okay, that means the total number of microbes living in you and on you is 10 times this range of values, 10 times 50 trillion or 10 times 100 trillion, right? So 10 times 50 trillion would be 500 trillion bacteria, viruses, protozoa living in us and on us, making up the circumstances that create our current status of health. Okay. All right, folks, I am out of time, and I think we've got a good start here in this chapter. I'm going to stop right now. Do you have any questions you'd like to ask? Okay. All right, folks. So have a good afternoon. We'll see you on Wednesday for lecture and lab. Take care of yourself.
Thank you.
Bye-bye.
Professor Todd.
Yes.
Hey, I just wanted to tell you.